So, we will continue with the discussion on this cytoskeleton structure a little bit more. So, this slide essentially shows that actin filaments, one is a schematic diagram on the right hand side of the slide. You can see that how these actin filaments are bunched together inside the cytoskeleton, and it extends throughout the cells, all through the cells. So, this is that top panel here in the left hand side, you see the actin filaments. This is the transmission under microscope image. Now, based on various structural characterization, both using microscope as well as the computational analysis. So, this is that helical arrangement of the actin filaments which are shown here. Now, if you look at the dimension of this actin filament, this is typically 25 nanometer. So, therefore, this one can be somewhere between 5 to 10 nanometer. So, this is the 10 nanometer around the diameter. So, it is extremely small, and if you consider the total cell size is 25 to 30 micron. So, there is a difference in terms of 10 to the power 3. So, that means you can immediately realize that so many large number of actin filaments can be accommodated physically within the cytoskeleton structure, simply because that cell size is much larger, it is around 25 to 30 micron, whereas actin filaments, the typical diameter is around 10 nanometer or so. Now, in terms of that their mechanical properties, which is important, because you remember that all these intermediate filaments, microtubules and actin filaments together, they provide mechanical support to, or provide structural mechanical support to various cellular organelles, and therefore, some understanding of their mechanical response, particularly under, let us say, under the application of tensile deformation or tensile force, that is necessary. Now, to start with, if you see that actin filament, their mechanical response, so it is extremely perfectly linear elastic response. The same is true for microtubules as well. However, there is a difference in terms of the two response between actin filaments and cytoskeleton, because this is here it is the force and this is here is a deformation. So, force versus deformation plot, microtubules has a much larger slope, you can see here, and actin filaments is not, it is a very small slope. So, therefore, elastic stiffness wise so there is a difference in terms of the elastic stiffness between the microtubules and actin filaments. The second point that must attract your attention is the way intermediate filaments they behave during this during the deformation under force, and you see that there is a very small linear region followed by largely non-linear region leading to fracture. And also in terms of the larger force, you can see, so both the intermediate filaments can sustain both in terms of large force magnitude as well as larger deformation. Whereas actin filaments, although the force wise it is the second, that is the maximum force it can sustain, but strain wise or deformation wise is much smaller than microtubules. Whereas microtubules, they can sustain larger strain, but at a extremely small force. So, this slide essentially captures the characteristic difference in terms of the mechanical response among actin filaments, intermediate filaments and microtubules. To summarize this particular behavior, these actin filaments and microtubules, they more show very perfectly elastic kind of response. Now, these microtubules shows elastic and viscoelastic type of response. Viscoelastic means, I will explain to you later. Viscoelastic means essentially it has a combination of both viscous fluid and elastic solid. So, these polymers, many of the polymeric materials, they essentially exhibit viscoelastic response. So, which is neither fully viscous fluid nor fully viscous solid. So, it is somewhere in intermediate between these two extreme behavior. Okay. So, now enough is about this cell structure description, and then we started with this normal cell structure description, and then I have explained that how this. So, to summarize, if you can see that couple of things that you need to so couple of things you need to remember for your own understanding, and these things essentially are that what is the typical cell size. So, that what is the cell membrane structure in terms of the bilayer phospholipid bilayer structure, double membrane structure, and in the cell membrane after the cell membrane what is the cytoskeleton structure like actin, microtubule and intermediate filament. And cell membrane structure also you have to remember that there are different channels like sodium potassium or calcium channels are there, and these channels some of these channels are voltage sensitive. Then second one is the cytoskeleton structure, this actin, filament and so on, how they are structured, what is the typical size of these actin filaments and so on. Third one that you have to see that what is the nuclear structure, and inside you have a DNA, that is the nucleic acid, and then also in the nucleus has a pore complex and pore the different things. And then fourth one is that how this eukaryotic cell structure is different from prokaryotic cell structure. Now, these all these 4 points as such I am trying to summarize here. These 4 points are sufficiently discussed in the last module as well as in the present module. Now, this cell has a unique structural characteristics that is now well established. Having said that, you have to see that how a cell can adapt to an external environment. So, cell can adapt itself. Now, this cellular adaptation process has manifestations in terms of the way the cells will also function on a material surface as well as how cells would respond to external stimulus. So, that is the reason I have made the first statement in this slide that various physiological and pathological changes at the level of individual cells or tissues or whole organs in response to prolonged exposure to cells to adverse or exaggerated normal stimuli is relevant to the cellular adaptation, which can be induced or regulated at any number of regulatory steps including receptor binding, signal transduction, gene transcription and protein synthesis. I will try to make an attempt, I will make an attempt to explain each of these things in subsequent modules, but to first discuss that what are the different ways a cell can adapt itself. First one is atrophy. Atrophy means decrease in cell size. Hyper means something larger. So, hypertrophy means increase in cell size. Third one is hyperplasia. Hyperplasia means increase in cell number. So, anything is hyper means higher. Metaplasia means change in cell type. So, example of change in cell type, for example, under certain circumstances osteoblast, I said in the last to last module, osteoblast can undergo transition to osteoclast. Osteo means bone, osteoblast means forming cells, osteoclast means bone resorption cells. Now, this kind of transition essentially helps the cell to adapt to certain external stimulation. Now, I might have mentioned in one of the earlier lectures that in a healthy human bone you have to have a appropriate balance between these two cells. In osteoporotic bone, like that is this large bone loss and so on, and or this osteoporotic bone you have a larger concentration of the osteoclast. So, osteoclast increases and therefore, that makes the bone osteoporotic. The fifth one is the dysplasia, that is a disordered growth in cellular shape, size or organization. So, this kind of cell shape or size there is a disordered growth, and this leads to the cellular adaptation, that is the dysplasia. Apart from this, the cells also can adapt itself in terms of changes in the cell shape. Now, how cell shape is changed? Let me take some time to explain these things in with certain details. Now, you have a cell here, Please follow me how I am making an attempt to explain. So, this is your actin filaments in the cytoskeleton, which you have seen more details in the last few slides before. Now, under certain circumstances, as I said, this actin filament has a unique characteristics that it can undergo the process called depolymerization. Depolymerization means that actin filaments will break down to smaller monomers, and these monomers are now distributed throughout the cell without any preferential space, without any preferential affinity to accumulate at a specific region of a cytoskeleton. But under certain signal or nutrient or a source, if that continues to happen, then these under favorable circumstances these monomers can get again assembled together by a process called repolymerization, and this process enables the formation of actin filaments again, but not at the same site where it originally got broken down to small monomers, but at a different site, and that way the cytoskeleton also can be stretched in that particular direction. So, let me reiterate my points once more. So, the process of depolymerization helps actin filaments to get broken down to smaller subunits, soluble subunits like monomeric units. Now, under certain circumstances, these or certain signaling environment, this repolymerization of that small soluble subunits can lead to again actin filament formation, not at the original place of their degradation, but at other sites of the cytoplasm, and that way this actin filament depolymerization and repolymerization can lead to the process called cytoskeletal reorganization. Now, this cytoskeletal reorganization is one of the precursor to cell material interaction. In fact, when a cell would try to adhere on a material substrate, try to adhere on a material substrate to protein-protein adhesion or protein-protein interaction, then what will happen? This, depending on some scenario, that cell would continuously change its shape and size. So, this I will address this point later, but at this point of time you might bear in mind that these changes in cell shape due to the depolymerization repolymerization is involved in the cell material interaction, and therefore, this has its own importance in the overall fate processes of a cell. Other things I have mentioned earlier, that is extracellular matrix and its importance in that overall cell fate processes. Now, this slide shows you little bit more details of this extracellular matrix. Now, what you see, this is your cell membrane, right, and in the cell membrane you see that there are something called transmembrane proteins, which I have mentioned to you before, and this is the transmembrane proteins is the integrins, and you have a plasma membrane here. Now, outside the cell, that means towards the other side of the cell membrane, opposite to the cytoskeleton or cytoplasm, this entire space where you have the different biological macromolecules and collagenous proteins are there. Now, this particular space is very important as far as the functionality of the cells are concerned, and this space is known as extracellular matrix. So, this is your intracellular region, and outside the cell is your extracellular region. Now, what are the various components which has been shown in that ECM? This is a very nice characteristics collagen fibers, and later on I will explain you that how this collagen is built up structurally. The other thing that has been shown here, proteoglycan molecule. So, these are like proteoglycan molecule or proteoglycan complex is there, and also you have other proteins called fibronectin protein, which are also present in the extracellular matrix region. And these are the summary of all the constituents of the extracellular matrix. You have collagen, that is the fibrous protein structure. you have fibronectin, laminin, these are also this other structure protein structure, then you have elastin fibers. Now, these two elements, collagen and elastin fibers, they give the elastic extracellular matrix the required elastic stiffness that it has. So, in other words, the presence of collagen and elastin fibers, they provide mechanical support to the neighboring cells that it contains. Proteoglycans, hyaluronan, and also this integrins and all, they are present. Now, integrin proteins, these transmembrane proteins has its own role, because these transmembrane proteins help in getting physically connected to the proteins which are adsorbed on a material surface. So, therefore, ECM structure and then transmembrane proteins are important the way a biological cell would interact with that of a synthetic material substrate. So, these are the different roles that extracellular matrix is expected to play. It provides mechanical support, it also controls the cell growth or control or maintenance of the cell differentiation, and it also establishes the tissue microenvironment. Many of these components of the extracellular matrix are synthesized or secreted by the biological cell itself, and it forms and it goes to outside the cell by a process called exocytosis. So, essentially this ECM is synthesized by the cell itself, and then it forms by certain biological processes, and it is a very nice network of both the different biological molecules, macromolecules, as well as different fibrous proteins. It also helps, extracellular matrix also helps in storing and presenting certain regulatory proteins also. So, for quite some time in the last module as well as this module, I have mentioned to you that this extracellular matrix contains proteins, as well as there are large number of protein molecules which are present. If you remember correctly, these are 10 to the power 9 number of proteins which are there in your eukaryotic cells, that is truly nucleated cells. You have around 10 to the power 6 proteins are there in a prokaryotic cells. So, these order of magnitude are important, and in view of the abundant presence of the protein in a cellular structure as well as that extracellular matrix, it is important for somebody who wishes to pursue this research or pursue this particular research area or pursue to study this biomaterial science or bone tissue engineering applications, that what is the structure of a typical protein. So, this slide and few more slides I will explain to you that how this protein structure is built up. So, this essentially substantiates what I have mentioned just 2 minutes ago. So, this is one of the most abundant intracellular organic molecules of biological polymer made up of smaller units known as amino acids. So, essentially amino acids is the monomers here in case of the protein molecule. In the context of the protein biomaterials, the protein molecules are the first one to adhere to a biomaterial substrates before it establishes the interaction with the cell surface receptors. Now, these are different biological molecules which are there. You have the sugar molecules, you have amino acids, and you have nucleotides. Then some of the proteins has a different shape, morphology, like globular proteins, and you have a very characteristic double helix structure of this deoxyribonucleic acid. And you also have a macromolecular structure like ribosome. If you look at the length scale, this is the 30 nanometer typical ribosome structure, and remember your cell size is around 25 to 30 micrometer. So, individual organelles is 30 nanometer, and if you remember that actin filamental structure which has a size somewhere around 10 nanometer. So, all the organelles and or all the cytoskeletal elements and different cellular organelles, mostly their size the length scale is in the order of nanometer. Okay. So, this essentially shows you that different level of structure description of a protein, but before that let me explain you that how this protein structure is formed or how this amino acid sequence is formed. So, you have this one of the amino acids. So, if I so this is how amino acid is written. So, you have a central carbon atom. So, you have a central carbon atom, you have a amide group. So, you have a nitrogen and you have a 2 hydrogen. So, this carbon atom has one organic radical, this is R, and one is this H hydrogen atom, and you have a carboxyl group here. So, carboxyl group means COOH, and this C essentially you can write it COOH. So, this is OH. So, in a physiological medium, these amino acids or the protein molecules in other protein molecules particularly is in charged conditions, it is not in neutral conditions. But just to show you that how this peptide bond forms, say essentially during the reaction of the 2 amino acids. So, the way amino acids are written is always keeping that N terminal in left side and C terminal is in the right side. So, when these 2 amino acids they react, then this will lead to one water molecule, and then you can start writing it from left to right. This is N terminal, this is C, this is H, this is R, this is carbon, and you have this oxygen, this is nitrogen, and this is H, this is carbon, this is hydrogen, this is R prime. So, you have one is that O, sorry, this is H, this is H. So, this is your N terminal. So, N terminal is always kept at the left, and carbon C terminal is always written at the right, or is always kept in the right. Now, other things that you must have noticed here, the 2 amino acids are different simply because if they have R or R prime are different, and the way these R and R prime, the way it is arranged, that forms is your amino acid sequence in a polypeptide molecule. So, essentially protein is nothing but a polypeptide, represents a polypeptide chain. Coming to this polypeptide chain again. So, in the protein molecules depending on whether you have this R prime, R prime, R double prime or different organic radicals are attached, a protein molecule has a different polypeptide, a different amino acid sequence, and therefore, 2 protein molecules will certainly be different. So, this has been shown here as I was explaining to you, and this amino linkage and peptide bond formation has also been shown in this slide, and this is what I was explaining to you, that OH and OH, and then this will be water molecules that will be released. Now, different type of bonds in the amino acid chains, it is mostly that weaker van der Waals bonds or hydrogen bonds are present, and to some extent in some of the cases you have ionic bonds are also there as is shown here. So, these bonds, one of the advantages of having these hydrogen bonds or van der Waals bonds in this protein structure is that if required, these bonds can be broken very easily. As a result, 2 polypeptide chains, they can be separated as and when it is necessary. So, this is the primary structure of the bonds that has been shown, and this is a typically helical structure, and it has a 5 turns in this helical structure, and it is typical dimension is 27 Armstrong, and each turn is 5.4 Armstrong. This slide shows more details about some of this where these hydrogen bonds can form. This is your polypeptide chains which are going in the major backbone chain, and these are hydrogen bonds that is between atoms have a peptide bond as an amino acid side chain that is also there. Now, as far as the stability of the protein molecules in a biological system is concerned, the proteins are more stable when it is a folded configuration. The moment it gets straightened, then it becomes very unstable. And one of the ways that so this is called denatured state of a protein. So, denaturing takes place when you use or you add urea to a protein solution, it immediately gets denatured. Now, protein structure, the understanding of the different protein structure itself is a discipline in itself, and then many researchers in conventional biological sciences, they do study the proteomics as well as the protein engineering and so on. So, people do lot of experimental study as well as they complement that with the computational study, just to understand the protein structure at different length scale as well as to understand the protein structure, how it evolves, what is the most stable configuration of a protein molecule, which is certainly beyond the scope of this module discussion. However, let me also tell you that protein structure at the higher level, it can be described by the warp modeling as well as the space filling model, and this has been shown here. So, now that you have some idea about this protein structure, the protein protein interaction is also equally important, or protein interaction with another biological macromolecule, that has been shown here. So, let me first explain this and then I will explain to you what is the relevance of that. One is the ligands, and this is your typical protein structure, it has a particular cleavage and then ups and downs. So, these ligands will come and will try to interact or establish the bonding with this protein structure, and these bonds are essentially non-covalent bonds. Non-covalent bonds means it can be either hydrogen bond or it can be van der Waals type of bonds. So, these non-covalent bonds are essentially extremely weak, and therefore, if these ligands will try to escape from these particular bonding sites, it is possible simply by breaking these bonds without much of the difficulty. So, this protein to ligand binding or ligand to protein binding is a precursor to understand that how a biological cell would interact with a biomaterial substrate, which I am going to describe in the next module.